Hi, my name is Nikita Hanley, and I will be presenting on the neuropathology of amyotrophic lateral sclerosis, commonly known as ALS. Throughout this presentation, I will be defining the characteristics of ALS, the onset of symptoms associated with the disease, its diagnosis, current animal models of ALS, the genes associated with the disease, the link between ALS and dementia, treatments, prognosis, and future research. ALS is a neurodegenerative disease that is associated with age and targets the motor neuron system of the body. The causes are poorly understood, and there is no specific diagnostic laboratory test. Neuropathological findings with classical ALS. It affects the upper motor neurons, specifically the cranial motor nuclei of the brainstem, and the lower motor neurons, the neural cells of the spinal cord. The upper motor neurons are responsible for the control of basic body functions, such as breathing, swallowing, and heart rate, while the lower motor neurons are responsible for muscle control. The degeneration of these motor neurons ultimately leads to the atrophy of striated muscles in the body. In a regular, healthy body without ALS, nerve cells in the brain, the upper motor neurons, initiate movement through the release of neurotransmitters. The signal is passed from the upper motor neurons of the brain to the lower motor neurons of the spinal cord. Nerve fibers extending from the spines, the axons, extend into the muscle, connecting at the neuromuscular junction. When the signal moves into the neuromuscular junction, it causes the muscle to contract, resulting in voluntary muscle movement. However, in patients with ALS, the affected motor neurons from the disease become unable to send signals to the muscle, resulting in the loss of voluntary movement and coordination, shown by the red arrow on the screen here. The onset of symptoms, meaning which symptoms present themselves first in the body, indicate the prognosis and the progression of the disease in patients. There are two main ways that the disease can present itself in the body, the first being limb onset and the second being bulbar onset. Patients with limb onset ALS experience motor neuron loss in the limbs as the first sign of symptoms. Patients with bulbar onset of symptoms develop dysarthria, difficulty articulating words, and dysphagia, difficulty swallowing. Despite the onset of symptoms, the main hallmark of ALS, or the main symptom associated with all patients, is the affected upper and lower motor neurons. To date, there is no specific diagnostic test for the disease. However, diagnosis is usually done by observation of symptoms. Accuracy rate of diagnosis is very high at around 95%. The most important or the most distinguishing symptom used for the diagnosis is multifocal motor neuropathy, which is described as the weakness and the atrophy of muscles. This symptom is mostly associated with the degeneration and the block in signaling of the lower motor neurons. Despite the high accuracy rate in diagnosis, it can be difficult to distinguish ALS from other neurodegenerative disorders. ALS and Kennedy's disease are very similar and are often confused during diagnosis. Kennedy's disease is known as a debilitating neurodegenerative disorder. The main differentiation of the two diseases used in diagnosis is that Kennedy's disease only affects the lower motor neurons of the body, while ALS affects both the upper and the lower motor neurons, as we already know. Due to ethical issues associated with studying humans, what is known about ALS comes mainly from studies on animals, primarily mice. Studies compared the principal features of human ALS neuropathology and those found in ALS animal models, and discovered that the main connection between the two was the presence of motor neuron degeneration and death at the cellular and molecular level. Animal models can be useful for studies of ALS in clarifying pathogenesis in humans and establishing reliable therapies and possible treatments. The main protein associated with the degeneration of the motor neurons in the body is TDP43, which is an RNA and DNA binding protein. The mutation TDP43 results in ubiquitinated inclusions in the gene. When a protein gets ubiquitinated, it gets tagged to die and will be destroyed in the cell. The TDP43 protein therefore leads to the mutation in the genes of ALS patients that causes their motor neurons and their cells to deteriorate and eventually die. The mutation in TDP43 is associated with the gene that encodes it, TARCPB. In patients with ALS, TARCPB encodes two pathogenic forms of TDP43 that cause excessive ubiquination in their body. Studies have discovered that several lines of mice expressing mutated forms of SOD1 a gene that encodes superoxide dismutase, developed a form of motor neuron disease closely resembling ALS. The researchers found that delivering a therapeutic interfering RNA molecule could suppress the expression of the SOD1 gene, improving survival of motor neurons in the brainstem and the spinal cord. It was concluded that in some patients with cases of ALS associated with the mutation of SOD1, the presence of an interfering RNA molecule could be of therapeutic effect for the treatment of the disease and could potentially improve survival of the motor neurons in the brainstem and the spinal cord. There is no current treatment at this time providing this technique. Dementia is often associated in patients with ALS, and it appears at the onset or later in these patients. The link between ALS and dementia may be associated with a mutation in a gene called CHMP2B. Little is known about the reasoning behind this pathogenic gene. However, studies have shown that two mutations in the gene, one that alters its function and another that causes low variations of the gene, are associated with the development of dementia in patients with ALS. These findings strengthen the link between ALS and the frontotemporal type dementia seen in those patients. Studies have found another gene that seems to be associated with the development of dementia in ALS patients, chromosome 9p21. This gene becomes pathogenic when a non-coding gene sequence, GGGCC, gets included into the chromosome 9p. Little is known about the mechanism behind this mutation, but research has linked this mutation to patients with TDP43-based pathology of ALS and who develop dementia. There is currently only one medication present for the treatment of ALS. The drug Riluzole is the only medication approved by the Food and Drug Administration for the treatment of ALS. It has been shown to slow the progression of the disease in some patients and reduce the damage of the motor neurons by decreasing glutamate release at the presynaptic terminal of nerve cells. Overall survival rate of patients differs with the onset of the disease. Patients with the bulbar onset have a shorter life expectancy, with 48% of them living between 2 to 3 years. Limb onset of the disease, however, is associated with a slightly longer life expectancy, with 24% living 3 to 5 years. Other important indicators for determining the prognosis of patients are the age of the patient at the onset of the disease, its early detection, the patient's rate of vital capacity decline, meaning how quickly the muscle associated with the basic life functions are deteriorating, and whether or not that patient is using Riluzole, the treatment for ALS. There are ongoing studies occurring on the use of stem cells for possible treatment of ALS. Stem cell research has led to the development of transplantation therapy. This transplantation therapy in patients with ALS may be beneficial for replacement of motor neurons already lost and could potentially counteract the degeneration and the death of motor neurons. And here I have listed all the references that I used throughout my presentation. Thank you.